So it's a great pleasure to reintroduce again Nikos. He told me that he felt that the presentation yesterday was too easy and too slow, so today it will go in much more detail. And he's going to give a different type of lecture today, telling us about the latest and greatest. So I'm not going to restate all the amazing scientific trajectory and bio. I want to just give two new things that I've learned in the last day or so about Nikos. One is that he's an amazing sailor. He can sail, and when he was here in Woods Hole, apparently he's so good at it that apparently he used to also help teach in the yacht club or in the sailing club, and help teach sailing. So that's one. And then the other one is I told you that in his undergrad he majored in music as well as math, and he's a very good piano player, and apparently he opened once for the Rolling Stones. Anyway, so with that said, let us welcome again Nikos.
Am I turned on? Okay, you can hear me now, right? Okay, thanks. Okay, thanks a lot. And indeed, yesterday, put it this way, it's okay? All right. So when Gabriel and Tommy told me if I could come and so on, I said I'd be happy to come, and I'd be happy to give half a talk, one talk, 22 talks, whatever you want, because I enjoy very much being here. And the one yesterday, because there were two talks, I thought it would be good for you if you are acquainted a little bit with all these methodologies that are not to be actually bought in the supermarket. You know, it's no matter what you do, whether you have something that you buy, an amplifier, or it's still all these things, they need adaptation to the experiments and to your questions. And there is a lot of technological stuff without which what you do sometimes is not 100%, safe, because you can have more than one interpretations, and you want to 2 or 3 or 4, and you want to reduce the number of possible. Now, today it's not going to be in that direction. I'll concentrate more on the results, but I will every now and then give you also a few definitions of things that we use on a daily basis, and it's not clear if everybody without certain type of biophysics or neuroscience, let's say, long education, would be aware of it. So, I'll talk about neural event-triggered fMRI. Yesterday, if you remember, we talked about direct electrical stimulation in fMRI. I thought that would be a very useful method, because you can selectively stimulate, whether you use electrical stimulation or you use optogenetic stimulation. You can selectively activate populations and then see the effects. And then if you want actually to compare stimulation or whatever else it is with the behavior, you don't just compare the stimulation of a particular site, but also what you see in terms of the activation of a whole network. Today, you saw also yesterday the limitations. There are certain problems that have also actually consequences for the application of direct electrical stimulation, as we said yesterday, in the case of the deep brain stimulation or many other things that people do. Now, today we'll be talking about something that does not require electrical stimulation, but you can exploit the fact that in each one of the structures of the brain, there are often populations, small populations or large populations, that generate local effects. People call them epochs or events. They're transient, phasic, or they are sustained, and so on and so forth. And you can see in the very next slide here, the time courses of electrical signals, broadband electrical signals that were recorded from different structures, from cortex, from PFC, from sensory cortex, from the hippocampus, from the thalamus, from pons, and from the parabranchial nucleus, one of the major sites where you have basically the propagation of the cholinergic activity in the brain. So you see these signals, and you see there are different deflections that in the beginning they may appear to be almost like artifacts. They are not. So there are some things here that we'll go into in small detail before we go into the interpretation of these things and the effect they have on global imaging. Before we do that, we want to answer one question. If you're aware of it, just tell me we know it and I skip it, I don't mind at all, is what kind of activity is captured by all these mean extracellular field potentials? People call them mean extracellular field potential because this broadband signal does not actually reflect the activity of 2.5 neurons, but it captures quite a bit of activity according to how close or how far different types of neurons to your electrode are, okay? So, one can ask, what should I understand when I see basically a time series like this one here or like this one here? And now what you see is a mixture of things. In the very simple case is the case that we all learned in the very beginning when we started recordings. Your electrode by chance can be extremely close to a neuron. Particularly the cortical cells, the huge pyramidal cells, they have very big bodies, and they actually have very strong currents that are going in and out during the activation of the dendritic sites of other types, basically of sensitive tissues. And if you are very close and the cell is activated, what you see, as you know very well, is basically this spike, as people call it, an action potential. And this action potential is considered to be a binary event. In most cases, you use the action potential sometimes to separate different types of neurons and so on. But in the end, you come to a point where basically this whole shape here of the depolarization, hyperpolarization, and so on is converted into a single line or a single dot. You have a binary function. Whether something happens or not happens is not so much of great importance. What is important is the rate with which things happen in the relationship of the spike sequences. There is, however, also cases, and this is in most cases, in most experiments the case, where the electrode is actually capturing activity that's much more global. It's not just basically close micrometers from a cell, but it's somewhere here and you have a lot of cells. If you have that, then you have to consider individual cells as dipoles. You have basically the neuron, the way I have it here in the scheme, and if the apical dendrites of the neuron are activated and input comes in, you're going to have very strong depolarization by the inflow of sodium, as you know, and outside basically of the cell, inside of the cell, you start getting actually more positive values. Outside of the cell, you're going to get negative values that you see. Then you have physics. You don't even need biology. It's Poisson's law. You have to have some kind of compensation. If you have a current sink somewhere, you have to have a current source somewhere else. And this usually happens in a distance. And in these cases, you see you have a dipole. And the moment you have a dipole, then in order for you to see approximately how strong the activation is, you have to compute the moment of this particular dipole. Now, what happens in most of the cases that we record from tissues, you have a lot of minuses close to the apical dendrites. I present here the simplest, beautiful case in cortex where things are simple. The moment you get into other structures, things become really messy. But let's say life is easy, and we have here all the minuses somewhere here, and we have all the pluses somewhere here. Then the sum of them, of the dipoles, it's going to be a lot of minuses here, a big minus, and a lot of pluses here, a big plus. And this sum is going to give you basically something that's representative of the activity that is not just 50 micrometers diameter in one dimension, but it goes basically to 500 micrometers or 1 millimeter or whatever it is. So the problem with the dipole summation, and to keep it in mind, because nothing is perfect, is that it's geometry dependent. Aligned dipoles or dipole layers have to be somehow clarified. Then it is based on knowledge of solid angle of each dipole, which you don't necessarily have in cortex. Again, it's ideal because you can consider almost all the pyramidal cells to be vertical to the surface of the cortex. And they are also based on the position of the electrode with respect to the dipoles and dipole layers. Now, what happens is you have two schemes. You can have all kind of combinations that give you the voltage or give you basically the activity in a particular area, but you can group them in a simplified version of it, so to speak. You can group them into two groups. The one is if you have the pyramidal cells that I said in cortex and in some other structures, you can have the same thing in some of the subfields of the hippocampus. Then you have this parallel organization, and you have all minuses in one more or less location and all pluses in one location, and this is called an open field generator, which generates very strong dipoles that are very well measurable and give you a very actually reliable activity profile for that particular spot. On the other hand, if you have cells like the stellate cells and many interneurons, where basically there are a lot of dendrites in all possible directions within a volume, then what you get is the plus minus here is going to be actually compensated by the plus minus here. And if you add all of them together, despite of the fact there is equally strong activation, you may end up having a teeny small dipole, and you think nothing is happening. This is what I told you yesterday when we were talking about fMRI. People criticize fMRI, but you have similar problems with electrophysiology. You just sometimes don't want to see it.
Yeah. But how often do you get input around the whole geometry?
Sorry?
In this picture, you get input symmetrically around the whole geometry, right?
That's correct.
How often is that true?
Oh, it's about 25 to 30% of the cases. A lot of structures like that. And there are actually studies, Murakami and Okada in 2006 is just one example. They measured basically the intracellular activity, where you measure the intracellular potentials, and you see how strong they are here, 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 and here. And they measure the current dipole moment, and you see they are very strong here because you have this arrangement of the pyramidal cells. The moment you start going to spiny cells or stellate cells, this becomes a teeny small thing. So that activity you lose. All right? So what that means, it doesn't mean you need antidepressants, it's all fine, but you have to take into account that when you're trying to understand the activity pattern somewhere, and in particular if you try to modeling, you have to have certain flexibility in the description of individual sources. Now, what people have been doing all along, and this is what the signals also that you saw in the beginning, they get the broadband signal nowadays. As you know, you can collect the data with 35 kilohertz or even with 65 kilohertz. You have very, very high in frequency domain resolution. You have all kind of small changes or large changes. And then you split them into two groups. One group is usually above 1,001 kilohertz. And this is what people call multi-unit activity. It mostly reflects, apropos, this is not a speculation. There are more than 20, 30 papers, some of them even 25 or 30 years ago, that they show very convincingly, indeed, that above 1,000, initially people did the mistake, they said above 500, but above 500 you have remote deflections that mimic the spikes, and they are not spikes. If you go into 1,000 kilohertz and you get basically the signal filtered, high pass filtered above 1,000, then you're going to get very, very good description of what people would call multi-unit activity. And the other thing is the LFP. Here I have it 0 to 600. You can just go 0 to 800, it doesn't matter. And this captures a lot of global activities. At the mesoscopic level, it captures a lot of activities that are very useful for understanding exactly perisynaptic events and in the cortex intracortical events. So the LFPs have been split into different bands. People call them the band-limited power signals, or BLPs. What you do is you filter, let's say, from 0 to 4, and then you get basically either the absolute value of the Hilbert transform, or you get the absolute value only. The Hilbert transform only works if the width of the bandwidth is relatively small. If you have a bandwidth of 20 or 30 hertz, do not even bother with the Hilbert transform, you get the absolute value, it reduces the error rate. So you get basically the delta, you get the theta, the alpha, the beta, the gamma, and so on. There are many other, every now and then there are new ideas about which band should be or could be important, doesn't matter. But the reason this is relatively important, the splitting, is because indeed by using some of these individual signals, you can much better describe the local activity in this relationship to behavior, or its relationship to other signals, like the signals we get with fMRI. Now, I go back to this one here, and you see, as I said, that these signals, the raw signals now, they may have spikes, and they may have also the LFPs at the very same time. They have funny things, the way you see here, the way you see here, the way you see here, and these things have been actually quite a bit studied and characterized over time, and they have specific names. What you see here, these up and down modulations, particularly in cortex, are named slow oscillations, and they are extremely pronounced during the slow wave sleep, during your sleeping, even during the deep sleep. And then you have the so-called K complexes, the theta oscillations, theta waves, the sharp wave ripples, the spindles, PGO waves, and so on and so forth. Now, some of these events, without even people having a much more general theoretical background, were studied extensively because they drew the attention due to their consistency and also to their origin, in which structure they usually appear, and also, as you're going to see, because they very early gave the impression that they are not a small local event, but something that reflects interactions between structures, sometimes structures that are far apart. So you see, for example, here the slow oscillations, they have a lot of structures that generate this particular pattern. First of all, you have the RTN or TRN, the way people call it, the reticular thalamic nucleus, which has a tendency to suppress the normal thalamic nucleus, so it stops somehow the sensory input. And during your sleep, this is of course important. As you know, your threshold is increased enormously. Somebody can come and do this to you, or you can have a noise, whatever, you're not going to hear anything. This is not because your brain has any problem, it's because the thalamic input is reduced to minimum. And then these neurons, they tend to suppress the sensory thalamic activity. Overlaying cortex mostly yields the oscillations that you see here. And these oscillations are remarkable because initially people thought, as I told you yesterday, in cortex a characteristic scheme of activity is this antagonism between excitation and inhibition. So initially people thought, in cortex something happens and you have excitation and inhibition going up and down, and basically that's why you see the oscillations. But when they first went and they started doing detailed studies and even intracortical studies, they noticed that both the excitatory and the inhibitory neurons, they go up and down at the same time. So the antagonistic level between excitation and inhibition does not change, but both excitation and inhibition, they go up and they go down and they go up and they go down, which is a completely new thing. It was a new thing, and it shows that there is another input to the sensitive sides of the neurons, along the neurons, that causes this modulation. And the thalamus is involved in the fine-tuning of these kind of things. So you have basically the cortex generating that with the help of subcortical inputs, and then the thalamus is also acting on this kind of rhythm and is doing the fine-tuning in a particular frequency domain. So then there are other things like the K complexes that I show you and the PGO waves. These were noticed a long time ago. Actually, the characteristic papers that I have are the ones that have been they've been cited a lot, and they are very descriptive and actually very good papers to read. But these are not the first papers. The same people have done work much earlier than 1997, showing that there are these events that can occur by themselves or in conjunction with other types of events. And you see here that there is this deflection. The deflection that you see in the signal can be 0.3 or 0.5 millivolts. It's a monster for the physiological signal. So it's a very, very large signal. And this signal can happen by itself, as you see here, the K complex, or it can happen in a way that is followed by these 10 to 18 hertz oscillations that are called spindles. So either they happen together or they happen always in certain transition states between the down and the up states that I showed you previously. And if you see at the right side, there are again deflections that are again at the level of 200 microvolts or 300 microvolts, and they happen during the REM sleep. People thought so, as you're going to see. This is not exactly the case, but they happen during the REM sleep, rapid eye movement sleep, and during waking very little, and they do not happen during the slow wave sleep, during the deep sleep. Now, these also effects drew a lot of attention because they thought they have something to do actually either with the consolidation of procedural memory, or they have something to do with the downscaling of activity. As you know, a lot of things during the sleep is being downscaled. You have downscaling of synaptic connectivity, and they drew a lot of attention. And another one is the theta oscillations that you see here. For example, in hippocampus, the theta you can see also in other structures, but it's very characteristic of hippocampus because it tells you the state of this structure and whether the structure actually is in a mode where information is being encoded and new circuits are being built, or it is in a mode where you have replay of activity and you have transferring of this replay activity to the cortex. So you have the theta, and now the theta, like all the other things that I told you, is also a perfect example of very long-range interactions. This theta activity here starts here in the brainstem with the nucleus reticularis pontins oralis. This is the NRPO that you see here, which suddenly, when in the awaking or attentional state or in the REM sleep and so on, what does? It starts firing for a long time consistently the way you see here, without any theta. This is sent to supramammillary nucleus, and it's a completely different structure, and there it is converted from a sustained response to this low frequency response from actually 4 to 8 hertz. All right? And does this continuously here, does this all this thing. The information from there goes to medial septum diagonal band of Procam, and there is fine-tuned, and then it goes to hippocampus, and it drives the hippocampus. Please.
You talk about information, not clear what exact information is being transferred from these areas. Like, what is the information exactly, and how is it quantified that's getting transferred from one area to the other? That was one of my questions.
The firing cells here project here, yeah, and the local circuitry here converts these sustained responses in because of very sharp intervention of the inhibitory neurons, these up and down states in the supramammillary nucleus. It was this oscillation. Neurons fire, stop, because of inhibition that is coming off local inhibitory neurons. And then they project here, and this is fine-tuned because there is a feedback also to the entorhinal cortex, many other things that we cannot go through without needing 5 hours. And from there it goes to hippocampus. And hippocampus under these conditions is in the mode of processing basically new information.
So that's where I was kind of confused. Like, what is the information? Like, what kind of stimulus is being represented here, or what exactly is this?
It's nothing else but a scheme that is used to basically synchronize different neurons with each other in hippocampus. It's a base, it's a time base that can be used. If you have a clock somewhere, you can say, okay, these neurons will fire every time you have a transition from the lower to the upper or from the upper to the lower. There is some kind of phase locking to this kind of oscillations that synchronizes neurons in the structure. Itself, it doesn't have any information about anything. But the whole thing, the way it develops, it generates the conditions that you have something that you can rely on to synchronize. If we are supposed to talk together for something, I say, okay, this guy is going to be doing tuk. When you start talking, please make sure you start talking when he does the first tuk, and I'll do also the same thing, and after that we are synchronized in some ways. So why? I don't think anybody knows. this is all over the place in the brain, and you have the same and the same and the same conditions where things need to be either with certain phase or completely synchronized or what. Okay? And finally, the final event is what I'll be talking about in the work that we have done together with functional imaging, is the sharp wave ripples. They were first reported in 1973, not 78, by John O'Keefe and Lynn Nadell in the first small paper. And then in the very well-known book, The Hippocampus as a Cognitive Map, they actually spent quite a bit of time to describe these events, because these events, as you're going to see in the next slide, they seem to have very strong relationship to the consolidation of memory. And they've noticed that they have two parts. They have a deflection, a very large deflection, like the K complexes or like the PGO waves, but on top of it, in the trough or in the peak of the deflection, there is a very fast oscillation that can go from 100 to 200, depending on species, 200 or 250 hertz. This is extremely fast, as you realize. And this oscillation and all the properties and the relationship of the oscillation to the actually deflection, to the sharp wave, and all these things were studied extensively by Yuri Butsaki in his laboratory. He did a lot of absolutely excellent studies describing what is happening, where things are generated, how it's propagated, what are the differences in the rodents between the dorsal hippocampus and the ventral hippocampus, and so on and so forth. Now, these events, as I said, have something to do with memory. That's why they drew enormous attention, and they have been studied intensively for many, many years. And before I tell you exactly what is happening in the relationship basically of these events to the theta oscillation that I talked about previously, I will remind you the basic scheme, the connectivity, the trisynaptic connectivity, as they call it in hippocampus. It's almost a one-way connectivity. It's very seldom in the brain that you don't have immediately loops and loops and loops. And what happens is basically you start from the top of the entorhinal cortex, from layers 2 and 3, and through the perforant path, the signal goes to the dentate gyrus, and then it goes with the mossy fibers to CA3, and the CA3, as you know, with the Schaffer collaterals, projects to the CA1, and from them to subiculum, parasubiculum, and other structures, and it ends up in the lower layer of the entorhinal cortex. Now, this loop has been known to be critical for a lot of things, including memory. And what happens is, the interesting thing is that when you have this theta, nothing happens in no ripple is supposed to happen, but the moment actually you have, the moment the theta is seized, you have immediate, basically strong depolarization in the CA3, very, very strong, simultaneous, and this very strong and simultaneous depolarization causes here a huge signal that you see in the CA1, and the CA1 has this excitatory inhibitory circuit, and it starts basically generating the very high frequency oscillations. And these are the ripples. Now, the ripples, as I said a couple of times, drew particularly attention because of their potential role in memory. And as you probably know, memory is supposed to actually work in two steps. In the very first one, information goes basically to the different cortical areas and also to hippocampus. Cortex, as you may know, is very slow in generating new connectivity patterns, and for good reasons. Otherwise, you would start learning all kinds of nonsensical things. And the hippocampus, on the other hand, is very fast in generating associational circuits, but it's also very fast in eliminating these circuits. So in the very first phase, you have this information going to cortex and going to hippocampus. And in the second step, which usually is supposed to be happening during the sleep and during the slow wave sleep, you have a replay of the activity of hippocampus that goes to cortex repeatedly, either in the sequence in which it happened during awakeness or in the opposite sequence, and is supposed to train the circuits of cortex for certain patterns and to add in the global, basically, formation of a particular sensory or associational area. Now, during the non-REM, the deep sleep, the sharp wave ripples coincide with the reactivation of neuronal assemblies, as I said, that were excited during our waking learning experience. The number of ripples increases after learning, and it predicts memory recall. In both rats has been shown, and in humans. Elimination of the sharp wave ripples during post-learning SVS interferes with memory consolidation, and during the awaking state, initially people thought this is only during sleep, but it turns out, Frank Loren and other people have very nice papers, several papers, they show that the sharp wave ripples, they occur at path choice points, potentially like a quick recall mechanism. So, with all this as background, it's not surprising that all these ripples, they drew a lot of attention, a lot of people trying to do experiments in humans, in rodents, or in monkeys. We are actually the first to do systematical studies in monkeys at all. And the question that we wanted to ask in this particular project, that was trying again to couple the electrophysiological recordings with the functional, with the bold fMRI, was what kind of multi-structure activity is related to the sharp wave ripples. The multi-structure activity, you'll see it again and again as actually MSA, right? Multi-structure activity, and I'll explain in a second what that is. With fMRI, the best thing you have is quite a bit of anatomical localization of structures, and if you have the right technology, this is very repeatable. If you know where to put, what to put, where the animal should be placed, and how to repeat your experiments, your little Raphael nucleus is going to be in the same position all the time. There is no problem at all. And you know exactly what it is, where it is, and you can go there and you can record from the nucleus, and you can verify basically what you see with the anatomical imaging or even with the functional imaging. So you have a lot of different regions of interest that here are sorted out alphabetically, and you can take the time courses of each one of these regions by excluding, in the beginning you don't make any selection of voxels, but you exclude voxels that have low signal-to-noise ratio. In each one of the images, very easily you can generate reliable signal-to-noise ratio maps, and then you exclude all voxels, no matter where they are below a certain threshold. And for each one of the regions of interest, the rest of the voxels are basically average, and they give you these time courses that you see here. The resolution of these time courses, of course, is lower than it is with the physiology. In physiology, you have basically data acquisition with 35 or 40 kilohertz. Here, if you use very fast imaging, as I told you, and different tricks that can be used with animals, your temporal resolution can be at the level of 500 milliseconds or 250 milliseconds, which still gives you, of course, very good signals. Now, to do this, we use the systems that I talked about yesterday. I won't spend any time. Most of you remember, these are the scanners, and the scanners were built in the lab. Basically, the magnet is coming from the company, and the gradients are coming from the company, but everything else was built in the lab by our own engineering group and the machine shops that we have. And here you have basically the state of the art today. You can have up to 5 chunks, and each one of these 5 chunks has 16 electrodes, so you can have 80 electrophysiological signals coming from different structures at the very same time that you have uninterrupted imaging. So the signals are acquired as if interference between the changing magnetic fields and the electrodes did not exist at all. Now, here are the first, basically, measurements within the magnet from the hippocampus. And as you can see, the ripples are very similar to the ones you would measure, of course, with rodents, or the few ones that people measure with humans. And you have this very large deflection that you see here, and on top of it you have this very fast oscillation. And it's amazing, if you see here the contacts that you have, a selection of the contacts, you'll see that in each one of them you have these patterns appearing approximately at the very same time, some of them actually in true synchrony. Now, if you get the profiles, when we started the study, I did not myself want to go into this a priori selective mode where you say, I'm going to filter the signal from 100 to 200, and then I'll take the absolute value of the signal, I'd set some thresholds and find, instead of that, to use some mathematically more appealing methods with which we did basically unsupervised clustering and detection of noise and detection of signals. And if you do that, in each one of the structures, you can determine how many local events you can have that are repeatable and robust, and you can rely on them and try to understand what they do and what the relationship between them is. And in this case, it was the hippocampal sigma and the hippocampal gamma and the ripples, as you would expect. The lower plots, we don't need to go into the details. They show basically the peak, the frequency of the peak of each one of them and the duration of them. And you can see that, as you expect, basically for the sigma events, you have higher duration, longer duration, and you have, of course, lower frequency. In this direction, this is the frequency range here. And as you go to the gamma and as you go to the ripple, the frequency increases. In the case of the ripples, you have something between 90 and 100 and 180 or 190 hertz. Now, this frequency depends on the head size because of the transduction, because of the conduction speed of the axons and the distance between different structures. So if you start with humans, you're going to see it's the lower possible frequency for ripples. It's not surprising because we do have bigger head than the monkeys. And if you go to the monkeys, then it's a little higher. And if you go to rodents, it can be really very high. They can have up to 250 hertz oscillations that are true biological phenomena and not artifacts or something like that. And now, what is characteristic, and it's very useful if you work with this because it's kind of signature of these events, is the phase coupling to the delta. All these events, they happen during the slow oscillations, particularly during the transition from the down state to the up state. And you see the peak here, approximately 90, 95 degrees consistently. Each one of these events, they're going to happen approximately in this transition from the low to the up state. And if you go into the details that people, to our surprise, had not done that before, probably because they thought it's not worth doing it, but it turns out actually if you compare these events with events that are happening in other structures, it's important to know these differences. There are different types according to the sharp wave that they have of different types of ripples, and the two ones here are the classical ones, and these are the less classical ones. And as you're going to see in a few slides, these different types, they have different contribution in the interactions between hippocampus and other regions like the pontine region and other neuromodulatory centers. Now, you have the event detection, and now you want to go to imaging and understand what are possibly the patterns, the MSAs, I call them multi-structure patterns, that have something to do with ripples. They are not produced. I said it very clearly. There's absolutely no causality. This is, as I said, a complex dynamic system. In complex dynamic systems, the emergence phenomena, they are due to interactions between the components. You cannot have one structure. You have the hippocampus and decides what the whole brain is going to do. It's a joke. You don't have things like that. So, and you have basically, what you want to do is you want to go and see basically when you have the ripples and see whether there are some patterns that are consistent around the time of occurrence of the ripples. And what you do, you have a threshold, and in the simplest case, you detect basically the ripples, or you use the method that I said before. It's just would take too long to go into the details of it. And you find the times of occurrence of the ripples, and then you take the signals, you generate a peri-event trial. For each one of the events, you have basically the whole signal around this event for plus, minus, whatever, 2, 3 seconds or 5 seconds. And then this signal is convolved with the hemodynamic response function. The hemodynamic response function, this a little bit to come back to the discussion we had about the way of interpreting the fMRI signal, is structure specific. There is no such a thing like a generalized. I know people wanted to do it, and I can understand their motivation. I would probably do the same thing. And Gary Glover had one hemodynamic response function that was using for everything he's using in every part of the brain. The intention is very well understandable, but the truth is basically that you cannot do that. Each one of the structures, depending of the local vasculature, of the connectivity, of the contribution of interneurons, and so on and so forth, it's going to have its own impulse response function. Hemodynamic response function is nothing else but the transfer function between one system and another system. The one system is, in this case, the neural system, and it has the neurons. They fire, they don't fire, they do what they do. And then you have the vascular system and all of the changes in the metabolic demands. And that particular system is not going to be identical because there are delays, there are slower processes, and it's going to be always some kind of impulse response function that characterizes the system. And the way to find what to do is to go into the systems identification techniques, and if you go into them, you'll see that there is selectivity, number one, and there are cases of non-causal systems. That means because of the feedback, there is no one-way, basically, characterization of something. So in all cases, now, in the simplest case, you have the hemodynamic response, you convolve this, and you get a regressor if you want to use the general linear model, or if you want to use any other, basically, nonlinear methods for trying to select voxels. And now, this is the response function. And if you take this and you run, in the simplest case, the general linear model, you'll see that during the ripples, there are some structures that appear to be up-modulated. They show a positive bold response. And some other structures, like the basal ganglia, cerebellum, tectum, and so on, they are down-modulated. They seem to have a negative bold response. Now, the way to deal with so many signals that represent actually different regions of interest in MRI is to forget about all the normal plots and generate something in the format of an image. So what you see here, instead of having basically positive for the hippocampus and the prefrontal cortex and so on, and you have these red or blue lines. This here means these, and this here means these, and the two blue, they means the two negative, basically bold responses that you see. From now on, you'll be seeing these images instead of seeing the plots, because we simply don't have enough space to put all these curves into one slide. And the second thing which was important was to understand what fraction of each one of the structure is up or down modulated. And if you see here, the structure is defined with a gray area, and sometimes only a part, as you would expect, of the structure is deactivated. The other part is not affected at all. And in some cases, there is a positive modulation, the way you see it here. And this is expressed in percent, and it gives you here basically the percentage of voxels in a particular region of interest that has been activated or deactivated. Now, you do that, and you get the first, basically, expression of what you would call a multi-structure activity during the occurrence or around the occurrence of the ripples. And this is much more comprehensive, and you can make better sense because you have the reds and the blues instead of having the curves, and you have here also the deactivated and deactivated regions, but still is not very well understandable. Go ahead, Nancy.
Yeah, so remind me, how long does a ripple last, and what is the temporal spacing of ripples? I'm just trying to think about how deconvolvable this is.
The temporal domain that you have is around, when you use the fMRI, plus minus 20 seconds. For each one of the...
But a given ripple lasts how long?
How?
A single ripple lasts how long?
The single ripple lasts 200 for 250 milliseconds.
And then are they bunched together?
And yes, we'll come to that in a second.
Okay,
I'll come to that, yes. I'll come to that in a second. I'll show you, because it generates a lot of complications. So you have here, as I said, the mixed blues and reds, and here you have basically the fractions that are activated or deactivated. And needless to say, I say that with pleasure, actually, because it just happened to be one project, so I was analyzing all the first data, and I was sitting in front of this and saying, my God, there's no way to understand anything. Look at what it looks, I don't know, we have to see what I should do. And then I realized a very simple thing, that the regions of interest here are alphabetically sorted. Alphabetically sorted for the brain doesn't mean very much, right? It doesn't want to know whether you call it accumbens or you call it whatever. And what I said, okay, then just to get maybe a better idea, what I'm going to do, I'm going to resort everything according to the ontogenetic, basically developmental categorization, where you have basically the metencephalon, mesencephalon, T-encephalon, prosencephalon, limbic cortex, and so on. So let's do this. And when you do that, suddenly, I see here basically what I just told you, the different sections of the brain that you could say, okay, I put in the brainstem these things, then I put the encephalon these things. And if you do that, here's what you get.